Our research project aimed to investigate differences in species abundance, diversity, and salinity within 5 different habitat areas around a salt marsh. To quantify diversity and species abundance of each habitat, we measured the oxic and anoxic layers of core sediment samples. Generally, if an ecosystem has higher biodiversity and abundance, it will have a larger layer of oxic sediment, as the higher amount of organisms disturb the sediment layer deeper and deeper.
We chose to study a salt marsh due to the diversity of biotopes contained within this ecosystem. We investigated 5 areas: in the salt marsh, at the edge of the salt marsh, next to a freshwater runoff, beside the rocks, and in the middle of the mud furthest from the salt marsh. We predict there will be a difference in species abundance as well as difference in oxic depth in our sediment samples.
A salt marsh is a coastal ecosystem in the upper coastal intertidal zone between land and open saltwater that is regularly flooded by the tides. Due to this, we collected our samples at low tide. The salt marsh, though at first glance may seem bare, is a diverse community home to many organisms, such as Grallata modesta, Amphipoda, and Terrabellida, as seen in our data samples. The great abundance of organisms in salt marshes provides food for larger creatures such as birds, which was evident from the many bird footprints we saw throughout the area. There are many biotic and abiotic factors that influence the sediment and species abundance in salt marsh areas. For example, we collected one set of samples near freshwater runoff in the salt marsh. This fresh water was rich in nutrients and fertilizers, and therefore caused the surrounding sediments to be very anoxic. Subsequently, the species abundance in these samples was relatively low.
To quantify species diversity and abundance in the relative areas, we collected 3 core samples from each area, as demonstrated here by Michelle. We then put those samples through a sieve to isolate the organisms and collected them for testing in the lab. We found the area with the greatest depth of oxic layer to be the salt marsh with an average oxic depth of 7.4 centimeters, and the area with the smallest depth to be the freshwater with an average oxic depth of 2.83 centimeters. The area with the highest species abundance was the rocks, and the area of the lowest species abundance was the salt marsh. As well, on the field we collected water from each area for the purpose of testing its salinity, which we did using the handheld refractometer in the lab. The salinity was found to be highest near the rocks and lowest near the freshwater runoff.